Hey guys, welcome back. Today we're going to talk about gel electrophoresis and PCR. This is a little bit of a longer PowerPoint, but hopefully you can keep up, and we will definitely go over any questions you have during class. Okay, so have you ever heard the saying that opposites attract? So let's talk about that. Okay, so we know that DNA molecules have a negative charge, and negatives will be attracted to positives, so a positive charge. So this is how we use gel electrophoresis. So agarose, agarose is a gelatinous substance, sort of like Jello, okay? And it's made by boiling red algae. And when you make it into a gel, it's smooth and sort of iridescent in color, but clear. But it has all these microscopic pores through which certain molecules can pass. All right. For electrophoresis, what you do is you apply an electric charge across a gel, okay, and so that the molecules have to move through the gel towards the charge that they're attracted to. So you have a terminal at the top and a terminal at the bottom, and you place the gel within it, and you usually place a liquid, sometimes water, but a lot of times it will be a salt water, a saline solution. The top terminal is negative. It will be black, and the positive terminal is red. I'm sure most scientists will tell you that one of the times they've done gel electrophoresis, they've gotten the terminals backwards, and it went backwards through the gel and did not work well, and it ruined all the results, which was a huge pain in the butt. Done it myself. You do it once and you never do it again. Okay, so what happens is you pour this gel, and then you put what's called a comb in the gel to make these little holes, to make wells. What you'll do is you'll pipette the DNA into the wells closest to, and those wells will be closest to the negative terminal. You'll then plug it in, and you'll turn on a power source, and the current will flow through the gel, and the DNA will be pulled towards the power source at the bottom. Okay, so here's a sort of an image of what a gel would look like. Up at the top, you have your wells. Okay, you have a ladder here. Okay, and some DNA is longer than others. So all the pores in this gel are the same size. You can change the concentration of the gel to make the pores smaller, but throughout the whole gel, the pores will be about the same size. What we know is that the smallest molecules will travel the fastest, and the largest molecules will travel the slowest. You run them for a specific time so that you get everything down to the bottom of the gel. You want to separate as much as possible. So when I was learning about doing gels, somebody explained it to me as if you were playing Red Rover through a forest, okay? So the number of trees in your forest is the percent of your gel, how concentrated your gel is. And the higher the concentration, the more number of trees, the smaller the spaces between the trees, okay? And then what's going to go fastest and what's going to go slowest, okay? You're running with a chain of people through the forest. So a chain of 10 people is going to take more time to run through the forest than a chain of 5 people, because those 10 people are going to have to go around more of the trees and bend and everything to stay connected, whereas the 5 people will be able to move a little more easily. I'll show you, and then we may do a little simulation with this, but what it comes down to is the shortest lengths of DNA will make it to the bottom of the gel, where the longest lengths of DNA will stay towards the top of the gel. Okay, and the length of the time will that you run it, you want to run it until your bottom part of the ladder, this is called your ladder of specific height, specific lengths, is at the bottom. Okay. A lane, okay, of course, a lane is the travel on the gel, okay, and the size ladder, or is DNA of known lengths, okay, and it's usually run in the first lane. Sometimes you will also run it in the last lane if you've got it something going all the way across your gel, just so that you can compare on both sides. But this is called your ladder, and you purchase your ladder to be specific lengths, and it's usually a unique color. Like here it's showing up as white where everything else is blue. Sometimes you'll choose something that's red, okay? DNA, we know, is a very small molecule. It's small enough to fit all the codes for every protein we could ever need into each and every one of our cells, okay? All right, that also means it's too small for a scientist to be able to analyze a single piece of DNA. All right. So to obtain meaningful information from the DNA, you must have a large amount of sample or many, many copies of a small amount of sample. Okay, so to be able to analyze it on that gel, that gel is not going to be able to visually see one piece of DNA. You either need to have a lot or a big chunk of DNA or a lot of copies of the same DNA. Okay, so in order to do that, we have two methods. We have RFLP and STR. Okay, all right, so RFLP, restriction fragment length polymorphism. So restriction enzymes are particular enzymes that cut DNA at certain sequences. Okay, it only cuts it at a specific cut site. So that's a specific sequence. All right, so it finds a specific nucleotide sequence. All right, and these cut sites occur with known places on the DNA molecule. Okay, but a mutation can change where the site will be, and it'll change the length of the fragments that it. So if you have a large amount of DNA, you can add a restriction enzyme, and it will cut the DNA into different pieces. Okay. The enzyme will digest the DNA, and it takes over several hours, and then you run it beside a gel, okay? And if the fragments on the two samples don't match, then they did not come from the same person, okay? So if you have, my DNA will cut at, say, space 3, 9, and 12, okay? And yours may cut at 2, 4, and 6, okay? We have different DNA. But if they found a DNA at on a site that cut at 3, 9, and 12, it's likely that it is also me. Now, that is not always true, but there's enough variation that with restriction enzymes that it'll cut pretty much the same for any one person, and you can eliminate enough people. RFLP is really time consuming. It can take up to 8 weeks to process one crime scene because you have to collect so much DNA for it to be worthwhile. All right, so what happens when we don't have a big old chunk of DNA? We do something called polymerase chain reaction. Now, the story I love to talk about with polymerase chain reaction is that the guy who came up with it, he came up with it driving on the Pacific Coast Highway while high on LSD. That does not mean you need to go out and do LSD, but it shows you the creativity that had to go into this. I believe he was a Stanford graduate student. He's also really crazy. He has written some very interesting stories about like crazy raccoons or something. So PCR is useful when there's very small amounts of DNA, and this allows you to replicate the DNA very quickly. So we isolate the DNA and we amplify it. So we rev it up and amplify it using polymerase chain reaction. We say PCR, okay? PCR, it approximates the body's own method of duplicating DNA to make millions of copies of a single piece that we want to study. So we're going to look at how PCR takes place. And this is the piece that the guy came up with sitting in his car driving down the Pacific Coast Highway doing LSD. So let's go back in time and see what he came. All right, so you have to have a certain number of ingredients to do this. First, you have to have a DNA sample, okay? Then you have to have polymerase. So polymerase, means SE, the ending means is an enzyme. Polymerase binds to open DNA and synthesizes a complementary strand. It is the way that DNA is copied in our body. In research we use what's called Taq polymerase, TAQ, Taq polymerase. All right, it does not. What's unique about Taq polymerase is it does not degrade at high temperatures. It is found in a bacteria found in deep ocean vents. All right, then you have these things called primers. Every primer is unique to the DNA sequence that you are looking at. And therefore, it's the specific sequence you're looking for is called the target sequence, and that is the piece you want to amplify. All right, and you have to have 2 of them, 2 primers. All right, and then you have nucleotides. Okay, this is your raw materials. This is your ATCG, your ATCNG. You have to have your deoxyribose sugar, and you have to have some phosphates, okay? Just like you had to have when you did your DNA model making. You cut them all out. Well, nucleotides, we mix them in, okay? And this is all done in a really, really small tube that's then put in a special machine. And the special machine is called the thermocycler. Thermo meaning heat and cycling. So thermocycle, it cycles the temperature. Now there are machines that you can set and you program them to cycle to specific temperatures at specific times. It used to be it had to be done by hand, and you would spend 30 seconds at this temperature, and then you'd pull it out of the heat bath and put it in a cool temperature, and then you'd move back. And it was very time consuming. People hated doing it, but it was the only way to get it to happen. Now we have machines to do it. Isn't that amazing technology? All right, and depending on your DNA, you'll get different time lengths, different temperatures. All right, so let's start with, we have this one strand of DNA, and the yellow piece is what we want to copy. Okay? All right, we're starting with 1 molecule of DNA. In reality, you usually have multiple molecules in a sample, but we're going to start with 1 and see how many copies we can make with just 1 little bit of DNA. All right, so first, the DNA is unzipped. Zippity, zip, zip, zip. Okay, by raising the temperature to 95 degrees Celsius. That's almost boiling. So it's going to open it up, okay? I could make a really bad joke there, and I'm not going to. But we take it to 95, and the DNA is unzipped. The high temperature, what happens is it breaks the hydrogen bonds that hold the nucleotide bases together. Here, we've unzipped it. We raised the temperature to 95. Zippity, zippity, zip. It's unzipped. And now we have 2 separate strands of DNA. All right, then we're going to lower the temperature to 60 degrees Celsius. Okay, what's going to happen is those primers that you've put into the mix to target that specific sequence are going to now bind to this open DNA. All right, these primers are then going to attract the DNA polymerase or the Taq polymerase to bind to the 3' end and to create complementary strands from the 5' to the 3' direction. DNA polymerase only works from the 5' to 3' direction. All right? Only DNA containing that target sequence, where those primers are going to bind, are the only places that are going to be copied. So at the end of cycle 1, there are 2 partially double-stranded DNA molecules here. So we've opened it up, pulled it down, and look, there's a primer attaching to that one. All right, and then the Taq polymerase comes in and fills it in. Primer tacks to the other one in the opposite direction and fills it in the other way. Okay, so now we have, after cycle 1, 2 pieces of DNA. And the blue is the primer, and then we filled it in with the Taq polymerase. All right, in the second cycle, we're going to open that up and unzip it again by raising the temperature back up to 95 degrees. All right, and then we're going to unzip it, like I said, at 95. Then we're going to cool it down to 60, and the primers are again going to match to the complementary strands, and then move in the 5' to 3' direction using the Taq polymerase. And at the end of cycle 2, we'll have 4 partially double-stranded DNA molecules. Okay, so looks like. Here we go. All right, that's after cycle. All right, the third cycle 3. We're going to repeat the first 4. We're going to take the 4. We're going to unzip them by raising the temperature, and then we're going to cool it down and attach the primers, and then allow the Taq polymerase to work. All right, the primers are going to create complementary strands with the Taq polymerase from the 5' to 3' direction. So if I keep saying what direction does Taq polymerase work? 5 to 3, 5 to 3, 5 to 3. Always remember that. All right, at the end of cycle 3, there are 2 double-stranded DNA molecules produced that contain only the target sequence. And we have 6 more that are longer all right, but contain the target sequence and some more. So let's look at, so we have 2 complete pieces after cycle 3. All right, and then we have some extended pieces here that have extra pieces. So the yellow is our target sequence, all right, and from the original we have 6 partial pieces, and then we end up with complete pieces. In cycle 4, the steps are repeated with the 2 doubles and 6 partials. At the end, there are 8 complete ones produced and 8 longer ones. So here we go. Now we have 8 complete ones and 8 partials. Fifth, 8 and 8 become 22 and 10. Okay, 22 double-stranded that contain only the target sequence and 10 that contain the partial. Here we go. After cycle 5, 22 and 10. And then we're going to go through this 30 times, possibly even more, depending on how big our sequence is and how many copies we want. About 30 cycles is normal. Okay, imagine having to do this, okay, by hand. But at the end of 30 cycles, you're going to have 60 longer length molecules. Okay, 60 longer length molecules aren't really going to show up on your gel. Okay, that's not a big enough piece. But the over 1 billion target molecules of this exact same length of your target sequence will be there. 1 billion of them will show up nice and clear on your gel. All right, 1 billion copies from one strand. In 30 cycles, I made 1 billion copies of a target sequence of a specific length. The 60 ones, we don't care about those. All right, the 1 billion, we can see. All right, so now, how are we going to use this stuff for us? All right, how do we use this DNA analysis for now? So here's a little thing. You're going to have to fill this in on your graphic organizer. So first, you have an event and then a transfer of material. You have identification, collection, characterization, DNA extraction, quantification, PCR amplification, detection, profile analysis, interpretation, statistical evaluation, and report. So let's go through each of these steps. First, the event. So during a crime scene, all right, various different things can happen, and biological material will be transferred from both victim and the suspect to items and surfaces at the scene. Okay, the types of the material tell us a lot about what happened at the crime scene, and they also give us a starting point for analysis. Okay, what are some sources of biological evidence? Blood, semen, saliva, urine and feces. Yes, there is DNA in your urine and feces. And your hair. Let's see if there's any other. Teeth and bone could also be there. Okay? Tissue. Say we ripped off some skin. And cells. Just basic transfer cells. Okay? All right, so let's look at identification, collection, characterization, and DNA extraction. So first, we have to identify potential sources of material. So they have to look at the scene and say, okay, where could we find some samples, okay? Then they're cataloged and collected. We talked about cataloging and collecting in our first unit. All right. Known samples can be immediately used for DNA extraction. So if we know something came from the victim or we know something came from the suspect, we can immediately send it to DNA extraction. Okay. Other things, we have to characterize it. Does it look more like this probably came from the victim or the suspect, or do we even know where this came? So that would require your step of characterization. All right. What happens after this? All right. First, samples must be analyzed to determine how much DNA is present. Can you use an RFLP? Do you need PCR? Is there enough there that you could run a urine test? Was there a ton of urine? Did just somebody wet their pants at the scene and you have plenty of urine that you could test for other things? Or do you have lots of blood and you could do some other sort of, like, could you start with doing blood typing and a class analysis and narrowing it down? Because some of those things are cheaper and more easily done than a DNA analysis. DNA analysis is very costly. Okay, PCR amplification increases the copy of the STR DNA. So you're going to choose for crime scenes, they're going to look at 13 different CODIS sections of STRs. So they're going to look at amplifying these tandem repeats. Certain sections of tandem repeats, and then you'll be able to, by looking at the lengths of the tandem repeats, then they can determine who it is. All right. Then the PCR products are going to run out on a gel. That's your detection. All right. And then they're compared and analyzed to suspects to either find a definitive mismatch or a possible match. Okay. So detection will be running out on a gel. Okay. All right. For each STR gene, we know the frequencies of each allele, and then we can calculate the likelihood a particular genotype exists within the population. So looking at all 13 of the CODIS locations, so there's 13 STRs that the government likes to look particularly at. They will find DNA and they will look at each of those 13 pieces. From those 13 pieces, they can generally narrow it down to a single person, or a pair of people, or a triplet of people, depending on if we have identical twins, identical triplets, and I think the highest we've seen is identical quads. Okay. From that, we have individual probabilities, and then and from those probabilities, create a report. All right. There are limitations, though. DNA is not the magic bullet for forensics, as much as it may seem like it. There are problems with DNA, okay? Depending on how a crime scene is handled, okay, the DNA can be contaminated, degraded, or even not present at all, okay? If it's done well. Also, there can be contamination once it's in the lab. When I worked in cancer research, one of the main jobs of a lab right by us was creating a system for finding out if your samples have been contaminated. If you've read anything about HeLa cells are notorious for contaminating things, and so you have to do continuous checks to make sure things haven't been contaminated. So contamination is a really easy thing, and it's happened plenty of times at crime scenes, and so DNA is not the magic bullet. Okay, there are also inhibitors that prevent PCR amplification from happening, which is a huge problem. All right, and you can also have DNA samples that mix between the victim and the criminal, and that can make them very difficult to separate when things get mixed up. STR genes are the same in identical twins, so further testing has to be done to distinguish between identical twins, but those tests can be even more expensive and time-consuming. So even though DNA seems like the 100% new frontier and nobody's ever going to be charged wrongly, it's not true. DNA is not without its own faults. So here ends the reading. I hope this was interesting, and I look forward to talking to you more about it in class.